You're listening to Conferences Online Allergy from Children's Mercy Hospital in Kansas City, Missouri. Today is January 8th, 2024. Our topic today is Abbas Chapter 4, Innate Immunity, presented by Dr. Nikita Raje. She is an associate professor of internal medicine and pediatrics and is the chief in the section of allergy immunology at Children's Mercy Hospital.
Started. Here are my disclosures, nothing related to the topic today. Learning objectives for today's talk are to recognize various components of innate immune system, to understand the functions of the innate immune system, and to recognize the types of reaction noted in innate immunity. Couple of pre-test questions here. Which of the following is not a function of the innate immune system? I'll let you guys go through that and then we'll come back to these questions at the end of the chapter. Toll-like receptors located in endosomal membranes of cells recognize which of the following? All right, so innate immunity. So what is innate immunity? It's an important part of the immunity that provides immediate response. The effector molecules of the innate immune system are fully functional even before the exposure or are rapidly activated. So some of the parts of the innate immune system are ready to go. Some of them are ready to be activated, but they are pretty quick. They give a quick response. There is no memory as such. So there might be some memory for innate immune system, but overall, there is no change based on that memory in how quickly it would respond to and how big of the response it's going to provide. So that consistently stays the same every time innate immune system is activated. And then the innate immune system has capability of recognizing only a limited number of molecular structures. And that's the base of how innate immunity works. The receptors of the innate immunity have different specificities compared to the adaptive immune system. So the same bug that's recognized by innate immunity, it might be a different part of that bug that would be recognized by innate immune system compared to the adaptive immune system. So the innate immune system has specific functions. Its job is to respond to microbes or to damaged cells, provide a barrier to the entry of those bugs, and then once it has recognized that there is a problem, it has to alert the adaptive immune system. It does this by two different ways. It either creates an inflammatory response or an antiviral response. So let's talk about some of these functions and reactions. So how does it respond to microbes and damaged cells? So the important part of that is the recognition of these structures is the first step, right? So the recognition of the molecular structures of microbes or damaged cells by specialized receptors. What are these receptors called? Anyone?
PRRs.
PRRs. Pattern recognition receptors, PRRs, there are multiple types of pattern recognition receptors, and their job is to recognize the specific molecular structures on microbes or on the damaged cells. So what are the molecular structures on microbes that are recognized by PRRs?
Like the PAMPs and the DAMPs.
Okay, so PAMPs and DAMPs. So PAMPs and DAMPs are pattern recognition. They are the targets of the pattern recognition receptors, and the overall response of the innate immune system is based on this recognition. There are, of course, some differences between innate and adaptive immune system. I feel like we have gone through these multiple times in over last few chapters. Just to sum it up, the innate immune system is specific for the molecular structure that could be recognized. Multiple different microbes can have molecular structures that can be recognized by a single pattern recognition receptor, compared to adaptive immune system, where there is a distinct pattern out for each of those microbe that will be recognized by a specific adaptive immune receptor. So there is limited number of molecules that can be recognized by innate immune system, molecular patterns or structures, compared to the antigens that can be recognized by the adaptive immune system. For the adaptive immune system, it's either the T cell receptor or the B cell receptor or antibodies that are used as the receptors, whereas we'll go into the details of the different types of receptors of the innate immune system. So on and so forth, we know that both adaptive and innate immune system have to have a way of discriminating between self and non-self. And that's how we make sure that the immune system only reacts to non-self molecules and not self molecules on the host cells. So the first step of the innate immune response is to recognize that molecular structure, the PAMPs and the DAMPs. PAMPs are the molecular structures that are shared by various classes of microbes. And these are typically structures that are essential for the survival of these microbes. When the cells are damaged, they produce DAMPs, right? They have these cells that are damaged or are dying due to any reason. They are gonna have these particular structures that can be recognized by pattern recognition receptors, and the healthy cells, in response to infections, can produce some alarmins that are picked up by these PRRs, so that they know that there is a problem going on and that there needs to be a response to those structures or molecular patterns. So pathogen-associated molecular patterns or damage-associated molecular patterns are the ones that are recognized by PRRs. So the PAMPs are, again, different types. So depending on the microbe, they may have particular structures such as nucleic acids, proteins, cell wall lipids, carbohydrates, and each of them is different in various types of bacteria, but they might be similar, or microbes, but they might be similar in a class of microbes like flagellin or pyelin or RNA or DNA. And then same with damage associated, so DAMPs, the molecular patterns can be different. These could be proteins or crystals, or these could be nuclear proteins. And because of that damage, these are the products, these are the patterns that will be produced by those damaging or damaged cells that can be recognized by PRRs. All right, so pattern recognition receptors, where are these? These are expressed by various cells. These could be phagocytes, dendritic cells, epithelial cells, other tissue cells, and all these cells have these receptors so that they can bind to the PAMPs or DAMPs that are in the that are coming towards these cells. So these PAMPs and DAMPs can be presented to these cells in various locations. So the PRRs need to be in various locations as well, connected to these cells. These could be on the surface of the cell, or they could be cytosolic, or they could be in the phagocytic vesicles, so endosomic. So they could be in any of these locations so that they can recognize PAMPs and DAMPs that are presented from various locations associated with the cells. But sometimes it's not the cells that come across the microbes. And so there has to be a way to recognize the molecules or PAMPs and DAMPs that are present in extracellular fluids. So there are some soluble molecules, not receptors associated with cells, but there are soluble molecules in extracellular fluids that can also recognize some of these patterns or PAMPs or DAMPs. And so those are pentraxins, galectins, ficolins, complement, and these are present in the extracellular fluids where they might come across the PAMPs and DAMPs. All right, so Let's look at the location of the pattern recognition receptors first. So we are going to go into the details of pattern recognition receptors. So these are all cell-associated PRRs. And then we are going to talk about the soluble molecules that recognize PAMs and DAMs. So cell-associated PRRs are various types. They could be on the surface, or they could be in the cytoplasm, or they could be endosomal. So looking at the list of these, toll-like receptors are one of the most talked about or most studied pattern recognition receptors. They are named from numbered TLRs 1 to 9 that are most studied. There are more that have been discovered, but some of these are on the surface. Some of these are endosomal. Whereas the NLRs, nod-like receptors, RLRs, rig-like receptors, or CDSs, we'll talk about these. These are in the cytoplasm of the cells. Then there are some other PRRs, such as CLRs, scavenger receptors, NFML receptors, and so various types of receptors that we'll be talking about. And then other than PRRs, there are these soluble recognition molecules that might be able to look at PAMs and DAMs and produce an immune response in the extracellular fluids. So let's start our discussion about the toll-like receptors. So toll-like receptors are present either, as I said, on the surface of the cell or in the endosomal membrane of the cells. These are numbered. TLR 1 to 9 are the ones that we'll talk about today. And so these toll-like receptors have a very specific structure. The structure is there is a conservative part of the structure that gives its name, toll. Toll was discovered as a Drosophila gene that is involved in the development of the fruit fly. And it was noted that the cytoplasmic domain, so the part that is in the cytoplasm of the cell, that domain of toll-like receptor is pretty similar to the receptor for cytokine IL-1. So it's called toll IL-1 receptor homology domain, or TIR domain, TIR domain. So this is very similar to what you would find in IL-1, IL-18, which have pretty similar structures as well. The outside structure, the extracellular structure that comes across the PAMP or the DAMP, again, has a very specific structure. It has, it's basically the ligand binding part of it. It has this convex structure and it has multiple leucine molecules, right? Amino acid leucine. And in between those leucines are various amino acids that form the ligand binding part of the TLR. So this structure is really important, and the way it gets activated is when it binds to the PAMP or to the DAMP, it makes a dimer, which means two molecules of the TLR will come close to each other to get activated. So it forms a dimer, and the dimer may have two different TLRs, like TLR1 and TLR2 coming together, so heterodimer, or TLR2 and TLR6 coming together, which are again heterodimers, but there are other TLRs which may form homodimers, which means two TLR2s or 5s can come together to form that dimer. So as you can see, each of this TLR has a specific PAMP or DAMP that it can attach to. For example, bacterial lipopeptides might bind to TLR1, 2, or 6. Peptidoglycans can bind to TLR2. TLR4 is very specific for lipopolysaccharide, or TLR5 specific for flagellin. All of these are on the surface. And this figure actually is really good to memorize, because this will kind of give you that visual memory of where these TLRs are located. TLR3, 7, 8, and 9 are endosomal, and they are specific for either double-stranded RNA for TLR3, 7, 8 for single-stranded RNA, and CpG DNA for TLR9. It's important to memorize these ligands that they bind to. One of the another part of this to memorize is UNC93B is really important for localization of these 3, 7, 8, 9 to the endosome. All right, so we talked about the ligands. One important part that is very specific to TLR4 is that it needs an adapter to bind to the LPS. So TLR4 is bound to MD2, and that MD2 also helps in that ligand binding. Again, remember, so MD2 and CD14 that are bound to the TLR4 ligand binding domain together form a structure that's important for binding to the LPS. All right, so overall, the reason for talking about any receptor is to make sure that you understand why these receptors, what are they responsible for. So going back to the structure here for TLRs, when they bind to these various ligands, the goal is that the receptor gets activated and performs signaling so that that signaling leads to the outcome that's important to counteract whatever they are binding to. So here, if the TLRs are binding to, say, gram-positive or gram-negative bacteria, the outcome of that receptor signaling should be that there are products out of that signaling that help to counteract that gram-positive or gram-negative bacteria. If it's a virus that these toll-like receptors bind to, the goal is that the receptors at the end of the signaling produce whatever molecules they end up producing are important for counteracting that viral infection. So as we know, innate immunity produces two types of responses. One is inflammation and another is antiviral response. So for any signaling, I'd like to start out with the outcome, so that the bottom of the signaling. So either we need antiviral state if these toll-like receptors are coming across viruses that they are binding to. So the PAMPs and DAMs are coming from viruses. The end result needs to be that there is an antiviral response. On the other hand, if it's another type of infection, then there should be inflammation. And sometimes there might be a combination of both. But these are the responses that the innate immune system has the ability to perform, right? So either inflammation, antiviral state, or antiviral response, or both. And to sum it up, the end result of that signaling is that it either produces antiviral response factors or interferon response factors called IRFs, or a transcription factor called NF-kappa B, and similar anti-inflammatory, inflammation-producing transcription factors like NF-kappa B that are responsible for inflammation. So the goal of the binding of the ligands to the toll-like receptors is to either produce NF-kappa B or IRFs. So keeping that in mind, let's look at the signaling. So all TLRs except TLR3 go through the signaling by binding to adapters and some other proteins. One important one is called MyD88. And the MyD88 ends up resulting into release of the transcription factor NF-kappa B. However, TLR3 and TLR4 can both go through this particular pathway that involves TRIF and TRAF, followed by activation of IRFs. So antiviral response. TLR4 basically can end up going in both directions, MyD88 and TRIF, to produce both inflammation as well as antiviral state. TLR7, 8, 9 can go through MyD88, but they end up in resulting into, again, producing both kinds of responses, antiviral response as well as inflammation. So these are things that you might have to memorize about the toll-like receptors. All TLRs except for TLR3 can go through MyD88. TLR3 only goes through TRAF, and then results into antiviral state, producing IRF3 particularly. Both the TLR3 and 4 go through TRIF. So TLR4 can go to MyD88 or TRIF. And then TLR7 and 9, even though they produce IRFs, they go through MyD88. So they cannot go through TRIF. They go through MyD88 response and then produce antiviral as well as inflammatory response. All right, so signaling, let's go through.
Hey, well, Dr. Raje, for TLR3, is it double-stranded DNA that it works on or double-stranded RNA? I thought the slide before it said double-stranded DNA, but I could be, I just couldn't flip back to the previous slide you were on.
Yeah, so it's double-stranded RNA, right?
Oh,
so
it's RNA. Okay.
Yeah,
Okay.
here it is. So double-stranded RNA. So think of the microbes that have double-stranded RNA in them, and that's what it's trying to respond to, right?
Okay.
So
Thanks.
thinking of TLR3 pathway, you know what it causes. So you know some of the microbes that have double-stranded RNA, and that's the PAMP, so that it can attach to.
Okay, appreciate it.
Mm-hmm. So DNA is only TLR9, right? CpG DNA. Rest is RNA.
Okay.
TLR3 is double-stranded, and 7 and 8 are single-stranded RNA. All right, so some of the adapter molecules that you have to remember is MyD88, and then that ends up in activation of a kinase. Kinase means it has some enzyme activity. That kinase is called IRAK4. IRAK4, MyD88, why are they important? Defect in them have known to cause severe bacterial infections, recurrent infections that may lead to life-threatening infections, including sepsis. And the issue with those immunodeficiencies is they don't end up causing inflammation, right? So there might not be fever, or there might not be elevation of inflammatory markers, but they can produce that significant infection that can be life-threatening. So kind of remembering these two molecules. IRAK4, as I said, is a kinase. It can end up activating some more pathways like the IKK pathway that we'll talk about again. And the IKK ends up, it's not shown here, ends up in releasing NF-kappa B, which is bound to its inhibitor. And once it's released, it can go to the nucleus where it can act as a transcription factor. That transcription factor, as the name suggests, helps in transcription of genes that are responsible for inflammation. So those could be cytokines, chemokines, they could be co-stimulatory molecules. All of them together produce inflammation. So that's the signaling for TLRs. On the other hand, the IRFs help in increasing. So IRFs will go to the nucleus and help in transcription of interferon genes. End result is secretion of type 1 interferons, which help in antiviral response. So TLR3 pathway defects, we have talked about this. All of these defects in UNC93, TLR3, TRIF, TRAF, all of those can lead to, anyone? What can it lead to?
HSV encephalitis. Is that what you're asking?
Yeah, HSV encephalitis. So, so that's a very specific presentation of toll-like receptor defect. All right, so toll-like receptors are tested a lot. You guys have to memorize that pathway, but also remember there are several other pattern recognition receptors. Again, to clarify, pattern recognition receptors are bound to the cell, and these are cytosolic, which means the pattern recognition receptors like nod-like receptors, rig-like receptors, or cytosolic DNA sensors are present in the cytoplasm, unlike the TLRs that we talked about, which were either on the surface or endosomal membrane. So nod-like receptors, again, the structure is really important because that leads to formation of a structure that ends up in the activation, leading to some of those signaling pathways or cascades, and end up in the end result of whatever it's trying to counteract. So the structure of nod-like receptors involves leucine-rich repeat. So the ligand binding is similar, but they have this domain called NOD or NACHT domain. It helps to interact with more NOD-like receptors. So that domain helps to form oligomers. So remember toll-like receptors where they form dimers with each other, NOD-like receptor forms oligomers. So not one or two, but multiple, few, not I should say instead of multiple, few of those NLRs come together to form a structure together. And then there is a third domain called effector domain, which can be different types. It can be C, so NLRCs, NLRPs, or NLRBs. So card domain, pyrene domain, or Burr domain. And so all of these nod-like receptors have two common structures, right? Leucine-rich repeat, which is helping in binding of the ligand. The nod domain, which helps to form the oligomers, so interaction between various NLRs. And then the effector domain is different for some of these groups. So card, or pyrene, or Burr. So card domain NLRCs, couple of them that are known are NOD1, NOD2. These are important in the GI tract. So they might be in the cytoplasm of GI tract cells. And when they come across specific structures, PAMPs, like bacterial cell wall peptidoglycans, they will activate the, activate the signaling. It recruits kinase. So remember in toll-like receptors, we talked about MyD88 and IRAK4. IRAK4 was the kinase that has enzymatic activity. Similarly, NLRs, they recruit kinases. One of them is RIP2 that helps to form a kind of a group of NLRs coming together called NOR signalosome, and that ends up in results into activation of, again, NF-kappa B. So that gives rise to inflammation. So NOD2 is present in intestinal Paneth cells, and NOD2 variants, gene variants, can lead to diseases such as Crohn's disease or Blau's syndrome. So if there is a loss of function variant, it causes Crohn's. If there's gain of function variant, it causes Blau syndrome. All right, moving on, let's talk more about these, right? So nod-like receptors, we know more than 20 different types. Here is the structure. So the leucine-rich repeat is the same in all of them. The nod domain or NACHT domain is the same, but when you look at, I don't know if you see my pointer, but when you look at the rest of the structure, it may differ in various groups. There is NLRA that has card as well, and another structure called TA. But then there are others like card, Burr, or pyrene domains. And so these, we talked about these here. Pyrene domain is the one that forms inflammasome and ends up in release of IL-1 or IL-18, and that leads to fever. So if there is a gain of function variant in pyrene domain, it can lead to periodic fevers. So here is that signaling here. So say there are these nod-like receptors called NLRP, NLRC4, NLRP3. When they come together, what happens is the nod domain brings various NLRs together to form a oligomer called inflammasome in this case. This inflammasome, bringing all these NOD-like receptors together with pyrene domain attached to it, is important to form inflammasome. This structure will activate caspase from procaspase 1 to caspase 1. And that caspase 1 is important in activating or pro-IL-1 beta or pro-IL-18 to its active form IL-1 beta or IL-18. This should happen when there is a signal that attaches to these NLRs. So PAMPs or DAMPs, right? So if there is an infection, then NLRP3 should get activated and form the inflammasome, and there should be release of IL-1 beta and IL-18, and that should give rise to inflammation as well as fevers. That's the reason, that's the protective function of these NLRPs. However, if there is a gain of function variant in NLRP3, it can lead to an overdrive of activation of caspase 1 so that it continues to release or cause activation of to IL-1 beta and IL-18, and continuous activation or continuous release of IL-1 beta that leads to fevers or periodic fevers. Apart from this IL-1, there are various other pathways in here that can get activated, or various molecules that can lead to activation of some of these NOD-like receptors, leading to autoinflammatory syndromes. So some of them, for example, for NLRP3 defects, it can lead to something called cryopyrin-associated periodic fevers or CAPS. These defects are Mediterranean of periodic fever, and those can be treated with anti-IL-1 therapy. So this inflammasome reaction is pretty important in the pathogenesis of some of these disorders. RIG-like receptors. So RIG-like receptors are cytosolic sensors of viral RNA, and once these are activated, they end up in the signaling cascade that leads to type 1 interferon release. RIG1, MDA5 are a couple of RLRs, and they recognize double-stranded RNA or RNA-DNA heteroduplex. So just kind of making sure that another type of pattern recognition receptor that's important in the cytoplasm. The last one is cytosolic DNA sensors. These detect microbial double-stranded DNA. They also end up in activation of type 1 interferons, and they lead to autophagy. One of the pathways is called STING pathway. Really important to know. STING stands for stimulator of interferon genes. As you know, this is a CDS, so which means that it binds to cytosolic DNA. Once it binds to it, it leads to phosphorylation of IRFs. So we'll see how that gets activated. Some of the other CDSs include DNA-dependent activator of interferon regulatory factors, IFI16, RNA polymerase 3, and AIM2. Let's go ahead and look at the STING pathway. So here is a double-stranded DNA. When that double-stranded DNA is in the cell, in the cytoplasm, we know there is STING. So this double-stranded DNA can produce a C gas that gets converted to cGAMP. That cGAMP can bind to STING, and that STING, when it gets activated, will give rise to activation of various pathways. So there's a signaling cascade that leads to IRF3 phosphorylation, and that's a transcription factor that goes to the nucleus and results in production or transcription of type 1 interferon genes. So that's how the antiviral response is produced by STING pathway. Defect in STING pathway, anyone? What is it called?
SAVI, right?
Yeah, SAVI. So Remember, these patients can have defects with STING pathway leading to SAVI, can lead to skin and lung inflammation. Give me a second, I'm gonna grab my water. All right, so what did we talk about so far? We talked about TLRs, NLRs, RLRs, and CDSs. Other PRRs include receptors for carbohydrates on microbial surface. These are called C-type lectin receptors or CLRs. These could be either on the cell surface or soluble. Here are some of the CLRs. So something to remember, VC sign or langerine. We have talked about langerine in the past. Mannose receptors, all of these are looking for specific carbohydrate that they can bind to. All right, other PRRs include scavenger receptors. These are on macrophages looking for lipoproteins. FPRs, these are looking for, again, peptides containing specific residues. All right, so that's the end of our discussion about how these PAMs and DAMs are recognized. Let's look at some of the components of the innate immune system. We know that there are these functions of innate immune system that includes just a barrier function, so physical or chemical barrier. We'll be talking about these soluble molecules that are in the extracellular surface, extracellular fluids, helping with recognition of PAMs and DAMs. We'll talk about cytokines and various cells of the innate immune system. Let's talk about the barriers. So the physical barriers provided by skin and mucosa. There can be a layer of keratin or mucus, cilia on the surface, and the peristalsis of that mucosa itself can provide a physical barrier. The chemical barrier is provided by the chemicals such as defensins and cathelicidins that are produced by the barrier. Defensins are produced by epithelial cells, NK cells, cytotoxic cells, as well as granulocytes. There are, in the GI tract, there are various types of defensins, alpha defensins and beta defensins, that help in disrupting the microbial membranes. These defensins can either be produced all the time, constitutive, which means it can be produced at all times, versus it could be increased or stimulated by various microbes or cytokines. Cathelicidins are produced by neutrophils and epithelial cells, and these also are stimulated by cytokines and microbes, and again have an anti-inflammatory role. There are some cells in the epithelial layer. These are intraepithelial T lymphocytes. So these are either alpha, beta, or gamma delta, but regardless of what kind of receptor they have, these T lymphocytes are part of the innate immune system, which means they have very limited diversity. They still help in killing microbes, releasing cytokines, activating phagocytes, but they are activated by very limited diverse antigens. So that's our epithelial barrier. Let's talk about the various cells. So we know that there are phagocytes. We'll talk about neutrophils and macrophages, and then there are dendritic cells. The dendritic cells have maximum number of pattern recognition receptors that we just talked about. The cell-associated pattern recognition receptors are maximally present on dendritic cells. There are also another type of dendritic cells other than classical dendritic cells, which are called plasmacytoid dendritic cells. These make the type 1 interferons that are important for antiviral function. They help in presenting the antigens to the naive T cells and help in activating them. Let's talk about the lymphocytes with limited antigen receptor diversity. We talked about intraepithelial lymphocytes. There are also invariant NK T cells. There are gamma delta T cells, there are B1 B cells, marginal zone B cells. All of these lymphocytes have very limited receptor diversity. Mast cells are present mostly in skin and mucosa. They have multiple pattern recognition receptors, like toll-like receptors on their surface, and they help in degranulation against some microbial activation. Innate lymphoid cells, these innate lymphoid cells are part of the innate immune system, which means they do not have gene recombination as you would see in the rest of the lymphocytes like B and T cells. So the ILCs also come from the lymphoid progenitor under the transcription factor activation called ID2. There's no rearrangement of the genes. There's no clonal expansion or differentiation, but there are various types of ILCs called ILC1, 2, and 3. ILC1 is a type of ILC that also has the name NK cells. NK cells are negative for CD3 marker, right? But they have CD56 and CD16. When we talk about NKT cells, all three markers would be positive. These are pure NK cells, which are ILCs. They make 5 to 15% of cells in blood and lymphocytes in blood and spleen. They are also present in liver, gravid uterus. IL-12 and IL-15 are needed for growth of the NK cells. 12, 15, 18, and interferon and gamma increase the cytotoxic activity and increase interferon gamma secretion from NK cells. The other type of ILCs are ILC type 2. They're similar to Th2 cells, so they produce IL-5, 13, help in defense against Th2 type of microbes like helminths. And then ILCs are Th17 type of, similar to Th17s. So ILC3s produce IL-17 and IL-22, help in intestinal barrier. Let's look at how the NK cells function. We know that they have perforin and granzymes that help in cytotoxicity. So when they come across a cell that's damaged by viruses or for some other reason, they will get activated and kill that cell, right? They also produce interferon gamma that helps in activation of other phagocytes like macrophages, and they help in bringing in other cells that help in killing those microbes. They also help in antibody-dependent cytotoxicity. So they use their CD16 that is present on the NK cells in this activity. They have various receptors on their surface. Some of them are activating receptors. Some of them are inhibitory receptors. So these are the types of receptors. So killer cell Ig-like receptors like KERS, C-type lectin receptors like NKG2D, CD16 or Fc gamma R3A, which helps in the ADCC. There are some inhibitory receptors such as NKG2A and some KERS, which are inhibitory as well. So remember, these inhibitory receptors have ITIMs at the end of them compared to the activating receptors that have ITAMs at the end of them. We had talked about ITIMs and ITAMs. We'll talk about it again if you guys need a refresher. These activating receptors recognize infected or injured cell, and they lead to the protein kinase activation, leading to phosphorylation of downstream signaling substrates. Inhibitory receptor, on this other hand, inhibitory receptor is supposed to recognize a normal cell. So if there is a normal cell that's not injured or damaged by microbes, in those cases, it will have self-MHC on it. That MHC is recognized by the inhibitory receptor, and it inhibits the NK cell from being activated. So if there is class 1 MHC on a cell, the NK cell will be inhibited and that cell will not be killed. Whereas if there is an infected cell, the typical virus will actually cause the MHC class 1 from not being expressed. So when that damaged cell comes across an NK cell, the inhibitory signal is not received by the inhibitory receptor. And the activating receptor is still active so that it results in the killing of that injured cell. All right, that was about the ILCs. let's talk about the soluble molecules. These are light pattern recognition receptors, right? These also look for PAMPs and DAMPs. However, they are present in the extracellular fluids. So they result in opsonization, promote inflammation, and recruit phagocytes, and they directly kill microbes. So the biggest one out of these is the complement system. We have talked about this. There are three pathways: classical pathway, alternative pathway, and lectin pathway. The classical pathway is dependent on antibodies. The alternative pathway is the default one, as well as the mannose binding lectin is the one that looks for mannose. So regardless of which pathway results in the activation, there is formation of a C3 convertase and then C5 convertase, which are the major steps of the cascade, and it ends into results into formation of membrane attack complex, which leads to the lysis of the microbe. So the classical pathway needs activation through C1q, C1r, and C1s. The alternative pathway is activated, it's spontaneous, and then it involves activation of or cleavage of C3, which is, as I said, it's the default, which means spontaneously C3 is being cleaved to C3a and C3b. Whereas in case of classical pathway, it's the C1 where the antibody gets attached to it and activates this pathway, leading to C2 and C4 activation. Those come together to form C3 convertase, and then that C3 convertase cleaves C3 to C3a, C3b, and then leads to the further cascade. Lectin pathway needs MBL, mannose binding lectin. Mannose binding lectin is associated with MASP and 1 and MASP2, so MBL associated serine proteases, and those together help in activation and cleavage of C3 to C3a, C3b. Here is C1q. This is the structure of C1q that is attached to two molecules of C1r. Here, small, two of the R's and two of the S. All of them together form this complex that can bind to an antibody complex attached to its antigen. When this C1, including C1q, C1r, and C1s, all of them get attached to the microbial antigen and the antibody, it activates the cascade that leads to cleavage of C3 to C3a, C3b. Similar to C1 along with QRS, the structure is very similar to mannose binding lectin. Here it binds to MASP1 and MASP2, and all of them together can bind to a microbe that has mannose in it. So this structure is looking for mannose, and any antigen that has, such as some fungi or some other microbes that have mannose in its structure, can bind to the MBL. Phycolin binds to N-acetylglucosamine, just like this MBL is looking for mannose. The phycolins can attach to any bacteria that have N-acetylglucosamine in it. And so they can fix these antigens and lead to a cascade that can help in recognition of these PAMPs. All right, so we talked about how complement system gets activated with C3 convertases. This is classical pathway C4b, C2a. This is alternative pathway C3b, BB, and that leads to cleavage of C5 to C5a and C5b. So this is C5 convertase, and that once C5b and C5a are created, C5b can help in binding to C6, 7, and 8. All of them together will form a structure so that multiple molecules of C9 get attached to it to form a hole in the microbe. Also, the smaller molecules or smaller fragments of complements can act as chemoattractants. C5a can also act as anaphylactoxin. And then there are various complement factors or regulatory proteins that help in preventing the alternative complement pathway from being activated so that it doesn't damage the host cells like factor H, factor I, TAF, MCP, CR1. Like complement, there are other molecules that help in recognition of PAMPs and NAMPs. These are called pentraxins, collectins, phycolins. Pentraxins are what we know as acute phase reactants, such as CRP, serum amyloid. There is also long pentraxins. These can bind to bacteria and fungi and activate complement by classical pathway. Collectins, like MBL, and surfactant proteins help in binding to PAMPs, for example, in the alveoli. Phycolins, they are looking for N-acetylglucosamine, and they can also help in recognition of some of the PAMPs. All right, moving on to cytokines. This is the last part of the chapter. We are talking about cytokines just of the innate immune system. So if you think about it, it's a long list, but if you categorize them that will help you memorize. So TNF, IL-1, IL-6, these are and maybe IL-18. These are the typical inflammatory cytokines. Type 1 interferons like interferon alpha and interferon beta are cytokines that are helping with antiviral response. IL-10 is one of the anti-inflammatory cytokine. IL-12, IL-15, these are needed for NK cell proliferation. IL-12 is needed for T cell response. We talked about IL-18. It has a pretty similar function as IL-1. IL-23, 27 mostly helps with T cells. So we will be talking about those. What do these cytokines do? They help in inflammation, local inflammation. From IL-1, IL-6, they help in activation of some of the inflammatory cells. They help in production of inflammatory markers like CRP. They also lead to systemic effects on various organs. And the end result of some of this might be sepsis or SIRS. Here's a classification of cytokines that helps you memorize. TNF, IL-1, IL-6 lead to inflammation. Th17 differentiation is from IL-1, 6, and 23. Th1 differentiation needs IL-12, IL-27 maybe, and then NK cells need 12 and 15. IL-18 as well, and type 1 interferons that also help in antiviral state. And IL-10 is one that has inhibitory response. All right, so talking about inflammation, we know that the inflammation is the end result of the response that is started by the recognition of PAMPs and DAMPs by PRRs. That leads to activation of some of these cells, mast cells, macrophages, epithelial cells. These cells then release cytokines that increase the permeability of the blood vessels. There is leakage of fluid, so plasma proteins, fluid, leukocytes that gets extravasated at the site and then ends up in overall response of acute inflammation, and over time can lead to tissue remodeling and chronic inflammation. Here is the acute inflammatory response from the injury on an infection that leads to these cells being activated, releasing of cytokines, and those cytokines leading to increased vasodilation and increased permeability. Some of these molecules being activated, complement antibodies, various other proteins that start killing those microbes. And then the leukocytes that come to the site of infection will lead to phagocytosis and killing of microbes. Don't need to go into those details. We just talked about these cytokines leading to inflammatory response. Now we want to talk about that response where phagocytes get involved and they help in phagocytosis. So quick word on that. Here are our phagocytes that lead to phagocytosis and end up in engulfing the microbe, bringing it in, and that phagosome binds to the lysosome to form phagolysosome. In the phagolysosome are the reactive oxygen species, nitric oxide, NADPH oxidase, that all of them together perform the killing. So we know phagocyte oxidase is one of the major important part of that response from the phagolysosome. NADPH oxidase helps to generate, along with its cofactor, helps to generate the act on the reactive oxygen species. And though that reactive oxygen species then interacts with superoxide, the superoxide uses dismutase to form hydrogen peroxide, and the hydrogen peroxide uses myeloperoxidase to produce bleach, so hypohalous acids or bleach. And the bleach or the hydrogen peroxide are toxic to bacteria, so are the reactive oxygen species. The oxidase acts as the electronic pump to generate electrochemical gradient. It increases pH and osmolarity, and that helps that's actually needed for the proteolytic enzyme action. So even though we think about NADPH oxidase as playing a major role, this electrochemical gradient also plays an important role in that overall reaction or response. So defect in this, we know, causes chronic granulomatous disease. Nitric oxide is important for macrophages. There is inducible nitric oxide synthase that converts arginine to citrulline and releases nitric oxide. Nitric oxide binds to hydrogen peroxide or superoxide to produce peroxynitrite radicals that kill the microbes. Also, these phagocytes have proteolytic enzymes like elastase and cathepsin G that also produces some of that effect. What are nets? Neutrophils that are bound to the tissue matrix. They are bound to them by MAC or some integrins. And when they are activated by microbial products, sometimes the neutrophils cannot phagocytose the whole microbe. And in those cases, it will extrude its DNA and granules onto that microbe to form neutrophil extracellular traps or nets. And that bacteria and fungi are trapped in it and then killed. So this basically shows that response from, say, a macrophage that has phagocyte oxidase, it has INOS, and it also releases cytokines that help in production of inflammation. All right, and the last one is antiviral response. The antiviral response is produced because of the virus infecting a cell, and that its components like RNA, double-stranded RNA, single-stranded RNA, DNA are detected by pattern recognition receptors in the on the surface of the cell, in the endosome of the cell, or in the cytoplasm. We talked about all these PRRs. All these PRRs, end result is they help in production or activation of interferon response factors or IRFs. Those transcription factors go to the nucleus, bind to the promoter regions, and lead to the transcription of type 1 interferons that lead to preventing other cells from being infected by the virus. So these type 1 interferons have paracrine or autocrine function and lead to kind of warn the other cells of the presence of the infection that leads to prevention of infection of the other cells or overall an antiviral state. And then the last function of the innate immune system is to alert the adaptive immune system. So the first signal is from the microbe where it gets activated, but the second signal that is needed for the lymphocytes to be activated is from the innate immune system that alerts them to the danger. And that's an important function of the innate immune system so that it can activate the adaptive immune system. There are multiple places where the innate immune system has its safeguards, such as IL-10 cytokine that helps in anti-inflammatory response, antagonists of or inflammatory cytokines like IL-1 receptor antagonist. This help. There is a way for autophagy of the genes that lead to autophagy in the cells, so that some of that inflammatory cytokines help in regulation of the inflammation that's produced. SOX stands for suppressors of cytokine signaling. They inhibit the signaling through cytokine receptors or through the STATs. And so JAK-STAT pathway, SOCs are antagonizing that pathway. And then SHP1 helps in TLR signaling regulation. All right, so a couple of post-test questions. If you guys want to go ahead and tell me which of the following is not a function of the innate immune system. I'll let you guys read through that. Anyone?
I'd say E, because we're talking about antibodies, so that's
Okay,
more.
yeah. Sounds good. Respond to fungal infections by inducing antibodies. Antibodies part of adaptive immune system, and toll-like receptors located in endosomal membranes of cells recognize which of the following?
Is this one A and C, nucleic acids and bacterial cell wall lipopolysaccharide?
So nucleic acids are the major one in the endosome, where the virus, where they'll come across the nucleic acids from various viruses, right?
Gotcha.
Okay. All right, questions.